Hi, everyone. My name is Justin. And for the past 8 weeks, I was working in Dr. Weinstein's lab under my PI, Dr. Ashley McDonough. And I'll be talking about my preliminary study on microglial activation in a piglet model of gut-brain axis inflammation. And to kind of give you a bit of background on our study, we have to go to the NICU, where it's found that premature infants have a high incidence of cognitive, behavioral, and motor deficits. And this is typically caused due to inflammation, which is also increased due to high susceptibility of infection and underdeveloped intestinal barrier. But it is also found that the diet of a premature infant can have benefits to the actual neurodevelopment, but the mechanism is unknown, which is what we study in this project. And this is also a collaboration with the Chen Lab at the University of Idaho, where we use piglet models. And the reason we use piglet models is because they are the best non-primate model, and comparing it to rats, it is better genomically, gastrointestinally, and the neuroanatomy too. And in that top right corner, you can see that the piglets, there was 2 sets of piglets, one for human milk and one for infant formula. And these piglets were fed over a span of a month or 28 days. And then tragically, they were euthanized. But in the name of science, they were then harvested. And the Chen Lab focused on getting the gut sample and blood samples, in which they studied the inflammation, the systemic inflammation, and how that affects neuroinflammation. And then we at the Dr. Weinstein's lab focused on how the effects of human milk reduces neuroinflammation. For my section of the project, we focus specifically on microglia, which are the neuroimmune cells of the central nervous system, which are a key indicator of neuroinflammation. And as you can see in the top right, that's one of the major indicators right there, where in the surveying section, you can see the highly ramified branch points in the resting area. And then once activated, these branch points retract, and then they turn into an amoeba-like state. They also release cytokines, but our main focus is proliferation, where we believe that infant formula group will have more inflammation, which would cause more proliferation of microglia and more activation overall. And in order to get the actual analysis, we have to begin by cryosectioning, which is taking those brain samples harvested from the Chen Lab, and we cut it up into thin little slices, place them on little slides, and then do immunohistochemical staining, which involves the primary and secondary antibody, where the primary targets the primary antigen, and the secondary is a little fluorescent indicator to find the primary antibody. And what we use for this is IBA1, which stains microglia, Chi67, which is for proliferative cells, and DAPI, which stains DNA. And in this image, you can see these colors in their respective antibodies. And in order for me to get this actual imaging, we go to the Mariana's microscope, and you can see the microglia in this image are red, which indicates that they are proliferating. And once I have these images, I get something like this, which is called a montage, which is a greater image of the actual microglia. And this is specifically the right prefrontal cortex. And I do something called serology, where I get a little mask of the area, and then I allow the computer to calculate random areas for me to do unbiased quantification. And all these little grids that you see in the top right are those zoomed in on the top left or the bottom left. And they have a little 3D plane in which I can scroll through and quantify all the cells. And with this, I found that after doing a t-test, that there was no significant status, but as you can see in the center graph, there were signs of general proliferation in the HM group, which could indicate that the infant formula could be lacking some nutrients or some key factor that doesn't allow it to be as proliferative. And in the third graph, you can also see that there is more proliferation of microglia in the IF group. And these dots indicate all the different groups that we use, HM1, 2, and 3, which indicate the piglets for each respective set. And in order to quantify this and have greater significance with our next steps, we can look at more serology, we can add more piglets to the actual sets, and we can also do morphology studies. And you can see in this skeleton that we will be focusing on the branch points and comparing that with other microglia and using that to find the indicator of neuroinflammation. And we can also further investigate mechanisms of how diets affect neuroinflammation. And with that, I'd like to acknowledge the Weinstein Lab for the mentorship and the support, the Chen Lab for the collaboration, and the Piglet Brains the funding. And I'd also like to thank Dr. Ellenbogen, Ms. Ellenbogen, Julie and Sylvia, Dr. McDonald, Jim Pridgeon, and everyone who supported me in this program. Thank you.